Hello, good afternoon, everyone. We will be doing lecture 4 today, the membrane transport. We will also talk about the lab next week, as well as quiz number 2, and probably have time for some review as well. So let's get started. Membrane transport. Let's do a quick review on the key components of a membrane, okay? So as you know from lecture 2, the main components of the cell membrane are these phospholipids, right? So phospholipid is a type of lipids, and it consists of one glycerol, two fatty acids, and one phosphate group, right? So the phosphate group makes up the hydrophilic head, which is shown here. Hydrophilic, which means it's water loving. So it will be facing the outside of the cell, right? This the stuff that's outside of the cell is what we call the extracellular fluid, or ECF. And this would be the inside, which is just the cytoplasm. Okay, and this here would be the cell membrane, right? If this is the cell nucleus, and we are looking at this here, if we zoom in, that's what we will see. Okay? It is the phospholipid bilayer. Phospholipid bilayer, because there are two layers of these phospholipids. So like I said, there is the hydrophilic head, which is a phosphate group, and then we have these tails. The tails are the hydrophobic. That means they are afraid of water, and they are on the inside of the phospholipid bilayer. And every now and then you would have like a protein that would, go through the membrane. If it goes all the way through, we call this an integral protein. Integral protein. Okay, and then sometimes you would have a protein just kind of like, stuck on one side, right? Either on the inside or on the outside. And these ones, instead of going all the way through, right? We call these ones, the ones that are stuck on one side, peripheral protein. Okay, and they all sort of have, different functions. Okay. What else is on the cell membrane? Well, sometimes you would have, well, not sometimes, like you would, every now and then you will have some cholesterol, right? That's the cholesterol. And the cholesterol is there to maintain the correct fluidity of the cell membrane. Okay? So by having some cholesterol there, it's going to make sure the phospholipid will not pack too close together. So your cell wall is not going to be, your cell membrane rather, is not going to be too solid. But you don't want too much cholesterol. Okay? Having too many cholesterol molecules will make the cell membrane too fluid, and everything would just leaks out. Okay. And sometimes on the proteins or on the phospholipids, you might have like, sugar molecules that are stuck on them. You might have like other, lipid molecules stuck to it. If a protein has a sugar stuck on it, then we call it like a glycoprotein, right? Glycoprotein. So an integral protein can also be a glycoprotein if there are sugar stuck on it. Okay, and you might also have like, lipoproteins that would be having a lipid stuck on a protein. Right? So that's like a really quick review of, some of the key components of the cell membrane. Now we are going to talk about the function of the membrane proteins. All right. So the first function, as shown in this picture, is that the membrane protein, and you can see this is the cell membrane, right? So this is the cell membrane, right? This is the inside, this is the outside. And one of the functions of the membrane protein is to attach to cytoskeleton, right? So we learned about cytoskeleton last week. An example of cytoskeleton would be things like microfilament, right? Or like intermediate filament, right? Or the other one, it's microtubules, right? So the membrane protein, right, provides support and maintains cell shape by creating anchorage point for the cytoskeleton. Okay. So like, you know, just to go back to our picture here, right? Like you might have some cytoskeleton here, right? So they need to attach to something. So there is a membrane protein that will help you, you know, anchor them, right? There's another one that will help them anchor them. It's kind of like create a scaffold, right? For the cytoskeleton to attach to, essentially. Okay, so why do we need microfilaments and intermediate filaments? Well, we need them to keep the cell shape, right? If you think about like a red cell, there's a little indentation in the middle, right? So to keep that shape, you would need to have some cytoskeleton for that. Okay, neurons, they look like this. They actually do. You just have to believe me for now. If you've never seen a neuron before, that's what a neuron looks like. And to maintain the starburst shape, you need to have the cytoskeleton. And those cytoskeleton needs to attach to something. That's what the membrane protein is for. Okay. If you have a question, please do not hesitate to interrupt me. Next function is the membrane protein can act as a receptor, okay, to pass down signal. Okay, so this is for cell signaling. Okay. The membrane protein, often an integral protein, acts as a receptor for we call them ligands, okay, for ligands, okay, which is basically things that binds to the receptor. And what could be a ligand? Example could be a protein hormone. Okay, so just to you know, give you a little bit of a quick lesson on hormones to help you understand this a little bit better. Right? So this is hormone, and we talk about this multiple times already. What are two types of hormones that we have in the body? Anybody? Please tell me. Two types of hormones. Yep, Christian gave me two examples of hormones. Very good, testosterone and estrogen, but I'm looking for two categories of hormone, okay? What are they made up of? Okay, thank you. So Megan said protein, so we can have protein hormones, which are like insulin, right? That's the example we use. And then there are the lipid ones, right? Which is like the testosterone and the estrogen that is mentioned. So those are steroid hormones. Okay. Now, steroid hormones is lipid, as I just told you, and the phospholipid bilayer is also going to have thick layer of lipids, hydrophobic tails in between. So the steroid hormones will have no problem crossing the cell membrane. So here is the cell membrane, here is the nucleus, and if I have a steroid hormone, it will just go right through, and then it will go into the nucleus, and then it will change gene expression. Okay, So we know genes are like DNA, and the DNA encodes for instruction to make proteins. So we can turn them on or off to make more of or less of a certain type of proteins. Okay, so steroid hormones will go directly into the nucleus, and then it will control the gene directly. Okay, because it is a lipid-based hormone, so it has no trouble crossing the cell membrane. On the other hand, if you have protein hormones, they are very big usually, and they won't be able to cross the cell membrane. So instead, what happens is you will need to have a receptor, okay, receptor, which is a membrane protein, and then the protein will bind to the receptor, and then it will cause a series of reaction to occur, and then ultimately that will change something in the nucleus. So this pathway is called a signal transduction cascade. Okay, it's just a fancy way of saying a bunch of reactions. Okay, so it's like, passing down commands, right? Like passing a note in class, right? So the protein is gonna cause the receptor to trigger one event, and that event leads to the next, and then the command just get passed along all the way to the nucleus. Okay, it's like this is one student sitting at the back of the room, this is sitting at the front of the room. There's no way to reach it directly, so you will reach it through a series of intermediates. Does that make sense? Are we together on this? Why are we even talking about this? Well, we're talking about this because of cell signaling, right? So this receptor here, that is the membrane protein. Okay, so let me write that down for you. If I can find my color. Right. So once ligand, okay, it doesn't have to be a hormone, it could be like a bunch of other things. Okay, once ligand binds to the receptor, a series of events, right? That's the signal transduction cascade, right? But it's really just a series of events will take place inside the cell. Okay, and that will lead to some changes, either in gene expression or cell behavior, depending on what the ligand is. Is everybody clear on this? Like what a ligand is, what a receptor is? I mean, you don't really have to use this word. You can just say like, things bind to a receptor, right? So like over here, this guy here, that is the ligand. Okay, some people pronounce it ligand. Doesn't really matter. Same thing. We good? Wonderful. Next, what's going on next? Well, another function is the membrane proteins. They can have enzymatic activity. Enzymatic activity. Okay. As you know, enzymes are proteins that can catalyze or speed up chemical reaction. Right? So a lot of times, many pathways involve, or we say many reactions involve multiple enzymes. Okay, so like you might start with the substrate, right? Substrate A, and then you will convert it into B, and then B will get converted into C. Right, and each of these steps involves a distinct enzyme. So you might need enzyme 1 to convert A into B, and then to go from B to C, you will need to have enzyme 2. So it makes sense to put these enzymes, number 1 and number 2, right next to each other so that the reaction can happen faster. Okay, like if this is not right next to number 1, and then the square will have to like find the enzyme before it gets converted to C. And sometimes you need the reaction to happen very rapidly near the surface of the cell. That's why the enzymes are arranged very close to each other. Okay, so enzymes belonging to the same path are located adjacent to each other, like next to each other. Okay, so this increase the rate of reaction, increase the efficiency of the reaction. All right. Moving on, we have two cells here. This is cell number 1. This is cell number 2. What happens is there's a membrane protein on one and then on the other, and these could stick to each other. Think of them as like little Velcros. Okay, when they're close to each other, they stick. Okay, and we call that formation of cell junction. Okay, so there are many different types of cell junctions or tight junctions, for example. There is like plasmodesmata, but we didn't talk about any of these. So we just call them cell junction in general. And the overall function of a junction is prevents uncontrolled movements between cells. Okay, so if you think about your intestines, right? So this is your intestinal cells, right? There are some wavy stuff called villi. You might have heard of that. You might not have, doesn't really matter. You'll learn it next semester. Okay, so this is like the inside of your intestine. That's where like your food is. You don't want the food to leak between the cells and go to, you know, who knows where. Right? You want the food to stay inside. So these cells, the intestinal cells, they would have membrane proteins that lock the cells together so nothing get past. Does that make sense? Very good. Next one we have cell recognition. Cell recognition. Okay, so this might be like a cell, and then this is like a white blood cell. Okay, so first of all, you should understand that there are many different molecules on the surface of a cell, any cell. Okay, your cell, my cell, bacterial cell, fungal cell, all the cells. Okay, so all these molecules that are found on the surface of the cell, collectively, we call them, right? We call them antigens. So how does your white cells, which are kind of like the cells that do the defense for your body, how do they know which cell belongs to your body and which does not? Well, they would check their antigens. Okay, if the antigens are same as all the other antigens in your body, then they will leave those cells alone. However, if they detect like a foreign antigen, all right, then the white cell is going to attack them. They might eat them. They might do phagocytosis. They might eat them. They might secrete chemicals to attack them. Okay, so how do they check for the antigens? Well, they would use membrane proteins to help do that. Okay, so white blood cells will attack anything that has a foreign antigens. Okay, has a foreign antigen on them. We'll say it has foreign antigens on them, which is why you cannot just, you know, take one person's kidney and then transplant it into the other without doing cross matches, right? Because the kidney is going to have different antigens than the person who's receiving it, and then the body will attack it. It will reject the organ. Any questions so far? Questions, comments, concerns? Okay. Last but not least, membrane proteins are important for transport. Okay, so charged or large or hydrophilic molecules or substances cannot pass through the membrane on their own. They required the help of a membrane protein. Okay, so those are the functions of the membrane proteins. All right, let's take a pause here for a moment. It seems like everybody who regularly attends the tutorial is already here. So I'm gonna go through what is going on next week. All right. So just before class, I posted an announcement. Yeah, so you can access all the tutorial recording here. Obviously, the one we're doing right now, it's not available yet, but it will be after class. And then it says complete lab 1. All right, so some of you might have done that already. And if you haven't already done so, I'm going to show you what is going on and how you can submit it. Okay, so I'm going to change myself to student view. So you just click on this one. And then it will take you to the lab page. Now, obviously, we cannot do the lab ourselves unless you have beakers and test tube and beetroots at home. But you can just watch the video. Okay, it's only 8 minutes long, and it's someone who's doing the actual experiment. Okay, take notes while you are watching the experiment because some of the questions in the lab are related to the video. Okay, and then once you've done that, you will have to download the Beetroot Lab. This is the one you have to submit. Okay, so you click on that, and then you will see this is the lab, right? So you cannot type anything in this lab. You have to download it first. So you scroll all the way down, and then there's the download button. Okay, so you click on that. It will go into your download folders. Okay, so you will open it. I'll click show in folder. There it is. And then you would open it. Enable editing if you have that little pop-up there. Okay, and then you would just read the stuff, obviously. And then you will answer the question, right? First one, what is the purpose of this experiment? Does anybody know what the purpose of the experiment is? Anybody watch this thing yet, or look into the lab? Anybody want to share so we can all get 2 marks? Or no, not really? Okay, so when you figured it out, what you would do is you would delete this thing, and then you would say the purpose of this experiment is to investigate blah 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 blah, and then you finish it. Okay, so that's 2 marks, and then you would answer it for all of the questions. Okay, so the first part has like 5 questions. I think it's out of 10. And then there are some questions about procedures, right? So you would fill it in, okay? Just fill it in. Make sure you answer all the questions. Now here, there is some data that requires you to draw a graph, okay? Last semester, like you would not believe it. Some people just draw the graph with hand and then submit it. And they are like my daughters are like 4 years old and 6 years old, and they could draw better graphs than that, okay? Compared to some of the stuff I see, it was just like terrible, okay? So please don't draw it by hand. Make sure you use Excel or Google Sheet. Google Sheet is free, right? If you don't have Excel on your computer. And I know you guys have learned how to do that from your computer class, right? If you don't know how to do it, you can watch the YouTube video. It's quite simple, but then you will plot that. Make sure you have label axes, proper titles, and you would submit your graph here. Is that okay? Anybody have any questions so far about the lab? Okay, and then you will answer all these questions. That's basically it. And then you will save a copy. Okay, you save the copy, and then you would go to submission. So how do you submit it? Well, you can go back to the homepage, and it says, and submit it in the assignment folder. So you press that. Okay, and you will have to scroll all the way down. This is just a rubric for me to mark. So like question 1, did you get 2 marks? Did you get 0.5, right? I will mark it. You don't have to worry about this rubric. You go all the way down, and then it says add the file. You click on that, and you say my computer, and you go back to the folder where you save your file. And you just drag it in, click add. And then if you have something you want to tell me, you can tell me. Otherwise, just leave the comments blank and click submit. And that's it. Okay, they will send, I think they will send you an email. Yeah, see a confirmation email. I just got the confirmation email, but when you submit yours, you will get it. Okay, if you didn't get a confirmation email, that means it didn't go through. Okay, you don't have to email me and ask me, did I get your assignment? Okay. Like you would get the confirmation yourself. So you just have to do this before October 8th, 10 p.m. Okay, but I suggest you hand it in as soon as you are done. Okay, and just get it out of the way. But sometimes you submit it and then you change your mind and you're like, oh, I want to make a change. Then you just submit again. Right? The new one will override the previous one. And then I won't start marking until after October 8th anyways. Is that clear? Everybody know what you have to do for the lab? No question at all. Are you sure? Okay, wonderful. Now, please don't look for answers on the internet. Okay, just watch the 8-minute video and then do it. It's much easier that way. Right? Some people go on the internet, find a similar lab, and then just copy the answers without going through the video, and the answers are usually not correct. All right, I've marked like I don't know, close to 1,000 of these. Throughout the time that I've been teaching. I told you I've been teaching for like 12 years, right, at Centennial. So I've marked a lot of these labs. I will know when your answer is from the internet because it will sound strange, okay? Like I would be like, we didn't even talk about this. Why would you say something like that? Okay, so just use your own words, right? Sometimes you might not get the right answer, and that's okay. As long as you can justify what your reasoning is, I will give you marks, right, for some of the discussion questions. Okay, so that's that. And another thing that's happening next week is, of course, quiz number 2. Quiz number 2 is going to be on Tuesday, and it will be available from 10 to 10, 15 minutes this time on lecture 3 and 4. Here's the link for it, and then you know, that's the number of questions that you will get. All right. In terms of difficulty, it's going to be similar to the first quiz. All right. Class average was not bad for the first one. I think it was 75-ish, which is quite high. Okay, but it's online learning, so it's expected to be around that range. All right. If no other questions, then we go back to this stuff. Now we have a diagram here, and we want to complete the diagram. On the left side, we have a bunch of macromolecules here. So these would be like food that you have ingested. Okay, so we'll call them macromolecule. Macromolecules from food. Okay, so let's say you eat you ate a burger, right? You will get some carbohydrates from the bun. You get some proteins and lipids from the meat. and maybe some carbohydrates from the vegetables. Okay, and maybe the ketchup is like sugary, right? So you would get some carbohydrates there. But we have to break the food down. We have to break down these molecules into monomers. Right? So if it's protein, we'll break it down into amino acids. If it's lipid, we'll break it down into, glycerol and fatty acids. So the reactions that we use to break down the food is what we call catabolism, okay, or catabolic pathway. Okay, because as you will learn in lecture 5, to break down glucose, it involves 20 steps, right? It's not a one-step process. There are many small steps, and each step is going to be catalyzed by an enzyme. So that's why it is usually called a pathway, right? Because there are many steps. So when you break down the food, you will be extracting the energy from the molecules, and you will capture all those energy, right, as much as you can to produce ATP, adenosine triphosphate, right? So that's the usable form of energy in a cell. Now, we as an organism is terrible in converting food energy into ATP. It's very inefficient. Okay, so we are only about 30% efficient, right? And most of the energy is actually lost as heat. So let me explain efficiency a little bit for those of you who find it a little bit abstract. Efficiency. Okay, what does that mean? Okay, probably the easiest way to explain this is to look at light bulbs. Okay, anybody still have these old light bulbs at home, right? As opposed to like the newer ones. What do we call these old light bulbs? Anybody know? What are those called? Nobody knows what those light bulbs are called? Or are you just not telling me? You know what I'm talking about though, right? Like the screw-on one, not the halogen one. The halogens are more like, sometimes you find it in like cars. Tungsten, yeah, the filament is made up of tungsten. Very good. This is the incandescent light bulb, right? Incandescent. Anyway, I assume you know what I'm talking about, these light bulbs. So you turn these on for like 10 minutes and then you try to touch it. How does it feel? It's going to be hot, right? It's going to be hot. So light bulb is very inefficient, okay? It's less than 10% efficient, right? So what does that mean? Well, let's think about what is the function of a light bulb. The function of a light bulb is to convert electricity into light, okay? If it's 100% efficient at doing that, you should waste no energy, okay? But it's not efficient because rather than converting 100% of electricity into light, most of the electricity is wasted as heat. That's why if you touch the incandescent light bulb, it's hot, okay? If you have one of these fluorescent light bulb, okay, the energy saving bulb, right, so to speak, you touch it, it's still warm, but not like hot to the touch, okay? So it's more efficient, right? Less heat is wasted. And then the really good ones are the LEDs, right? Okay, if you have LED lights at home, you touch it, it's not even hot. Okay, LED looks like the traditional one, but then it's frosted on the surface. Okay, so LED. Right, so those ones are very efficient. So as a person, our efficiency is roughly 30% or so. Okay, 30% to 40%, depending on how you calculate it. Most of it is wasted as heat. Okay, it's not all bad though, right? Like we need the heat, right, to keep us warm because we have to maintain a body temperature of 37 degrees. So the heat is for that. Okay, and then what do? Well, we're going to take these monomers, right? These are building blocks, right, for making molecules that we need. And to build things, it would be anabolic pathways. Okay, so what are some of the things we need to build? Well, maybe we need to make an enzyme. So we need to take amino acids and build a protein. Okay, maybe we need to create some storage form of energy, right? So we would have to build glycogen, and maybe we need to repair the cell membrane, and we have to make some phospholipids. Whatever the case, right? Taking the monomers to build a macromolecule, we call that the anabolic pathway, and that requires ATP. And then in the end here, we'll have macromolecules needed in the body. So essentially, the key thing here to remember is energy released from catabolic reactions or catabolism is used to drive anabolism. Right, so as long as we're eating, we'll be able to create ATP, and we'll use those ATP to allow the anabolic pathway to occur. Any question on this? Just a quick word about ATP. So as I told you, ATP is adenosine triphosphate. So there is an adenosine here, and then there will be 3 phosphate group with a very energetic bond at the end. So this is ATP. And then what happens is you will break the last bond by adding water. It will be hydrolysis, just like before. Then you will get the ADP plus a phosphate. And in the process of doing that, you will release energy. Okay, and then you can use this energy to do different things, such as building molecules. But then now you would have an ADP, adenosine diphosphate. That's not very energetic anymore. You need to recharge this molecule. So to recharge it, you will have to supply energy. Okay, it's like your phone. You got to plug it in, recharge it. Right, so where does the energy come from? Well, the energy comes from catabolic pathways, right? So catabolic pathway, breaking down your food, that will provide energy, and then you will lock that energy, use that energy to make ATP, okay? And then you'll use it, and then you'll remake it, use it, remake it. So you can keep on doing this again and again until you stop eating, okay? Until we die. That's basically the cycling of energy between ATP and ADP. So what do we use the energy for? 3 different kinds of cellular work that requires ATP. Well, we can use the ATP to power things like mechanical work. Mechanical work. Okay, so what's mechanical work? Such as movement. Movement of cilia or flagellum, right? Or muscle. All those things. Anytime you move anything in the body, you're going to need to have ATP. So that's called mechanical work because you're physically moving something. We also need ATP for chemical work. Right? So just like we mentioned earlier, for anabolism, right? Building molecules, you need ATP for that. And then the last thing is transport. Okay? So active transport. If we move things from an area of low concentration to an area of high concentration against their concentration gradient, then you would have to supply energy in the form of ATP. So those are the 3 kinds of cellular work that requires ATP. Next, 4 factors that affects the rate of diffusion. And you help me out here. What are the 4 factors? Maybe list some factors that you remember. You can unmute and tell me. Cynthia says size. That is correct. Okay, so smaller molecules equals to faster diffusion, faster rate. Anyone else? Anything else besides size? Yeah, there's also charge. Okay, so let me explain charge a little bit. If you have 2 parts here, there's a membrane, and then you have a bunch of H plus here. Okay, and then maybe some H plus here. So the H plus will be driven to go this way because of 2 driving force. Number 1, there is the concentration gradient. Okay, so that's going from high concentration to low concentration. Number 2, there is an electrostatic gradient. Like charges repel each other. Okay? If you have a lot of positive charges close to each other, they don't like to be that way. So they will push away from each other as much as possible. So that pushing force will cause some of the H plus to move away. Okay, so if the charges are like charges, then that will speed up diffusion. Right? If you have both positive and negative charges here, then let's say I throw some chlorine in here or something, right? So the chlorine will now be attracting the H plus, preventing them from leaving. Okay, so the charge could either speed up or slow down the rate of diffusion, depending if it's like charge or different charges. Christian said temperature. Yes, temperature is definitely another one. So generally speaking, high temperature equals the faster rate of diffusion. Okay, think about last time you were in a classroom, which could be a long time ago. You know, if someone is wearing perfume or cologne, you would be able to smell it much quicker on a hot summer day compared to like a cold winter day. Okay, and then finally, the last one, Cynthia says steepness. So that is the steepness of concentration gradient. Okay, so gradient just means the difference, right? So think about like a ball rolling down the hill. Okay, so if it's not very steep, it will still roll down, but compared to very steep, this is going to roll down faster, right? So the greater the difference between the height of the starting point and the end point, the faster it's going to roll down. Now, in terms of concentration gradient, let's say there is 70% salt solution here and then 20% salt solution. So the difference between the left and the right is 50%, right? And the salt will go this way. Now, let's say we have a 70% here, but 50% here. It will still go to the right, but because the difference is not as great compared to the first case, the rate of diffusion will now be slower. Okay, so the steepness is basically referring to how much of a difference are we talking about between 2 points? Is it a big difference or is it a small difference? The bigger the difference, the faster the initial rate of diffusion will occur. Okay, eventually it will all be the same. Okay, but you know, initially one will be faster than the other. Are we okay? All right, so let's do a quick poll question and see what's going on. Polls. Okay, go ahead. Select all correct answers. Okay, so let's see, this is catabolic reaction. That means breaking things down into smaller pieces is the stuff that is on the left side of the diagram we labeled earlier. So does it consume more energy than they produce? No, it doesn't. It has to produce more energy than it consumes because we need to get those energy, right? And trap it in ATP. Excuse me. Break down polymers into monomers. That is correct. Produce excess heat to warm the body. That's also correct because of the inefficiency that we talked about. It does not only occur in human cells, it could occur in plant cells and other cells as well. So that is not true. And so the answer is B and C. This one we will come back. Okay. Let's talk about another one here. This one, it's let's say we have a tube, right? We have a tube here. Okay, a U tube, a U-shaped tube. Okay, U-shaped tube. I'm gonna put a membrane here, and then I'm gonna dissolve a bunch of solute here. Okay, so red balls, solute. Right? And we're gonna have the water. The whole thing is filled up with water. And at the bottom, we are going to have a membrane selectively. Okay, we'll just say only allow water to pass through. Okay. My question is, what will happen over time? What will happen over time to the water levels? So A, no change. B, left goes up, right goes down. C, right goes up, left goes down. And then D, both go down. All right. Let me give you the poll and you let me know. Okay, 10 people have answered and we are neck to neck right now. There are two answers, 50-50. Okay, just wait for a few more response and then I will stop. Anyone else? We need 5 more people. Is it because you guys don't see the polls or what? What's the problem here? Maybe they're just away from the keyboard. Okay, here we go. So which is it? Right? Half of you who've answered say is B, the other half say is C, and then one person said no change. Does anybody want to explain? Like, can someone who chose B, one of the 5 of you who chose B, would you justify your answer for me? And similarly, if you chose C, can one of you tell me why you chose C? So, you know, we can convince each other why it is B or C, or maybe it's A. Anybody? No? All right. So the correct answer is B. Okay, and here is why. First of all, the red dots, they cannot cross. because they're too big, right? It says only water can pass through. So water will always go towards the side that will dilute the solute. So it's a lot of solute here, which means the water will go this way to try to dilute this. Okay, and as water goes through, then the right side level will drop, and then the left side level will go up. More importantly, okay, listen carefully. More importantly, what does that achieve? By dropping the water level on the right, now you have the same amount of solute, but in less volume. So that's going to cause the concentration on the right side to go up. And then by adding more water on the left side, you will be diluting the content. Okay, so the water will stop moving when the concentration on the right and on the left are equal. Okay, so you will be concentrating the solution on the right side, but it will be diluting the solute concentration on the left side. So Christian asked, what is the solute again? It could be anything. It doesn't really matter. Okay, I said salt at the beginning, NaCl, but it could be proteins. It could be a bunch of other things. Okay, as long as it's dissolved in the water, that's what's going to be driving the movement of water. So this is called osmosis. Osmosis, which means water will move towards the more concentrated side. Concentrated side. Is this okay? Does everybody understand? Those of you who chose C, do you understand why the answer is B? This question is on the quiz, okay? So make sure you understand everything I said. I mean, I cannot guarantee you're going to get it, but a question like this is in the bank. So you might get it. You might get it. All right, let's move on. Now we have three situations. We're going to put a red blood cell, which is what this thing is, into different solutions. Right? We have the isotonic solution, the hypo, and the hypertonic solution. So let's take a look at what's going on in each cases. So for the first case, if it's isotonic, it means the concentration of solute, right? Concentration, square bracket means concentration. That's the official symbol. Okay, so concentration of solute inside equals to concentration of solute outside. Okay, so you can't really see the picture too well here, but like you would have the, same amount of dot distributed on the inside and outside. Okay, if it's like 70% solute on the inside, it would be 70% solute on the outside. So what does that mean? It means the rate of water moving in to the cell is going to equal to the rate of water moving out. Okay, so like you would have the same amount of water moving in as you would out. Okay, maybe I'll change this to green arrows, easier to see. And you will have the same rate of water moving in as out. It does not mean water is not moving. It just means that they're moving in the same way as moving out. So no change. Okay, no net water movement. No change in cell shape. Okay, another way to visualize this is if you have a bucket, and I put a hole at the bottom. Okay, so there's some water in the bucket. Okay, and then it's leaking like that. The same time it is leaking, I can turn on the tap. Okay. If the rate at which the water leaks out is same as the rate at which the water is coming in, then what happens to the water level? No change. This is exactly what's happening. The amount of water that's coming in, like from the tap, is same as the amount of water that's leaving from the leak. So no change in the cell shape. All right. So this is if it's an animal cell, this is a happy cell. Happy cell. Cell is happy. Okay, that's what they want. They want isotonic environment. Next, we have hypotonic. Hypotonic means there are a lot more solids on the inside. So you can add these dots. Makes it look like a poppy seed bagel. And then you have less outside. Okay, so solid on the inside is greater than. This symbol means greater than. You learned that in math, I think. And compared to the solid outside. Lots more dots. So remember our discussion with the U tube, right? The U-shaped tube. Which way will water move? More water will go in. More water will go in compared to out. Okay. So the rate of water moving in is going to be greater than the rate out. So that's like, you know, turning on the tap more. So more water comes in like that, and then it's more coming in than leaking. Then the volume will increase over time, right? So net movement, net water movement equals to into the cell. And then cell will swell. And if it's really severe, might burst. Okay, so bursting is called lysis. Okay, or we say the cell will lyse. Okay, lysis or it will lyse. Is that okay? No questions at all? Okay, thank you. Next, we have hypertonic. Oh, like I probably mentioned this already, like the hypotonic, right? Like you see the big O here, that's how you can remember. Hypo means the cell swells up and it's gonna go kaboom. Hypertonic is opposite. You have all the dots outside, all the solids. Okay, you have some inside, but not as much. Okay, so solute concentration inside is less than solute concentration outside. As such, you will have more water leaving. Than coming in. And so this time is like you drill a bigger hole, right? And then you have more leakage compared to the faucet, right? So over time, the rate in is less than rate out. And you have a net water movement out of the cell. Cell will shrink, and we call this plasmolysis. Okay, I don't know if I talk about this in the lecture, but you have a part of the brain called the hypothalamus. Okay, hypothalamus. You will learn that next semester, and then you know there are like blood streams surrounding it. Okay. And what happens is if you're dehydrated, if you haven't been drinking your water, if you're sweating a lot on a hot summer day, then your blood concentration is going to go up. It will cause the cells in the hypothalamus, which is deep in your brain, to shrink. The shrinkage of those hypothalamic cells is going to create an electrical signal that goes throughout your brain, and it makes you feel thirsty. Okay, and then you drink the water, you dilute your blood, and then the cells swell up again, then you won't feel thirsty again. Does that make any sense? So there you have it. Hypertonic, hypotonic, and isotonic. And that's 1:30. Let's take a break. Right? When we come back at 1:35, we will continue. All right, let's keep going. Next, we have to compare the passive and active transport. Okay, so on the left here, we have the active transport. And in terms of the movement of molecules, active transport would involve going against the concentration gradient. Okay, so what that means is it will go from low concentration to high concentration and going uphill essentially. And in order to do that, you will need to use ATP. So requires ATP from the cell. Can someone give me some examples of active transport, please? Anyone at all? Anybody? No? No examples of active transport? That's okay. So we can have things like exocytosis. Okay. We can have things like sodium potassium pump. Okay. If you're not sure what those are, please watch the lecture recordings. For passive transport, it is going down the concentration gradient. So going downhill. This is going to go from high concentration to low concentration. Does not require ATP from the cell. Maybe something else is supplying the ATP or it doesn't require ATP, but it's not going to need it from the cell. Examples of these passive transport include diffusion, osmosis, okay, something also called facilitated diffusion. Okay, so facilitated diffusion, just to refresh your memory, is when the molecules cannot cross on its own because maybe it's too big, maybe it's charged, maybe it's it's polar, then it needs the help of a membrane protein to cross the cell membrane. And that is what we call facilitated diffusion. There are two types, right? There is channel protein or the carrier protein. Okay, back to the poll questions. Let's try this one. Okay, more people participated this time, which is good. And most of you got it right. It is B, right? Movement of substance down its concentration gradient. Let's try another one. Let me move my picture away. This one is select all correct answers. So I will give you this one. Go ahead. Okay, these are the results. Let's see. When a cell is placed, it's a hypertonic. So hypertonic means that the cell is gonna shrink up, right? Because you have more solid inside. Okay, so next movement of water out of the cell. No, it's gonna be into the cell. Okay, and will it burst? No, because you are. Oh, sorry, I drew it wrong. My bad. It's gonna move out like this. Okay, net movement of water will be out of the cell. That is the correct one. Okay, and will it burst? Nope, because it is all the water is leaving, so it will actually cause it to shrink. Okay, plasmolysis. The cell will only burst if it's an animal cell. No, it will not burst because water is leaving. And why is the water leaving? Because the solid concentration is higher outside compared to the inside. So the correct answers are A, C, and E, which are the majority of the answers that you guys have chosen. Very good. Okay, next one. This is one answer, so go ahead. Okay, this one is interesting. I am afraid nobody got the answer for this one. So make sure you know the answer for the quiz. All right, the correct answer is small hydrophobic. Right, remember your cell membrane has this thick hydrophobic tail regions, right? So this whole lipid region basically creates a barrier for anything that's charged or hydrophilic. Right? So in order to cross the membrane, it has to be hydrophobic. Remember what we talked about earlier with the protein and the steroid hormone? The steroid hormone can cross directly in, no problem, because it's lipid based, right? Okay, so for it to cross easily, right, easiest, it has to be hydrophobic. And then you ask yourself, is it easier to cross if you're big or small? Well, obviously it's going to be small, right? Because you would have an easier time squeezing through. So the correct answer is C. Does that make sense? Have I convinced all of you that the answer is C? Or are there any follow-up questions? All right. All right, now I have an application question. I tried this with my other classes and they kind of like it. I will see if you guys like it as well. Now I'm going to give you a link so that you can see the question even after I move away from the website. No, that is not the link that I want. Let me see where it is. It is this one, right? Let's see. Yep. Okay, so I'm gonna paste it in the chat. Can you guys click on the link in the chat right now? Chat box. Go ahead, click on that, and it should take you to this page. Okay, can you do that, please? Give me a thumbs up or some kind of signal that you've clicked on it. And then I'm gonna put you guys in a breakout room, right, to discuss about the question. There are two questions. I want you guys to talk about it. I'll give 5 minutes or so to talk about it, and then we will come back and hopefully, you will be able to give me some, some of your findings. Okay, so let me see. Breakout room. Okay, so when I put you in the breakout room, the person whose name is closest to like alphabetical order, the person who is highest in the alphabetical order, you will read the question to the group, and then you will start the discussion there. Okay, let's hope there is no awkward silences, but we will see. Okay, how many people do we have here? We have like 16 people. Let's go with 5 groups, so we have about 3 people per group. Okay, and go. Right, so you will get an invite, and then you can join the group. Once you join the group, you won't be able to see my screen anymore. That's why I sent you the link. What was that? Did that work for you guys? Did you enjoy having that little opportunity to discuss, or it was just awkward silence? Do you prefer to just do the polls? Let me know. Okay. Well, I don't think you guys will talk, so I will just launch a poll to see what you think. Okay. Okay. A, you like that, Okay. You want to do more. B, stick with the polls only. Okay, polls only. C, no preference. Okay. Okay, it's good. Christian said we got some discussion done. That's excellent. Okay. All right. Okay, well, overwhelmingly, most of you say you prefer to just stick with the polls. Well, it was worth a try. Okay, I understand not everybody is comfortable sharing. It's not like the classroom. Classroom, I put you in group and then I, kind of breathe down your neck, right? I hover over each group and make sure you're discussing. Right now, I cannot enforce that. But for some of you who had a good discussion, good for you. That was good. Now comes the results part. Anybody want to share some of what they discussed in the breakout room for this particular question? What is the correct answer for the first one? And for those of you who do not want to share, you can just choose the answer that your group has agreed upon for the first question. What is the answer? Good. The water will flow from the beaker into the bag, causing the bag to increase its volume. That is very good. You know, more people got it right than when we did the polls individually. So maybe that's because you guys had a chance to talk about it. Maybe not. I don't know. Let's find out the next one. Nobody got it right in my other class for the next one. Let's see if you guys are the better class, so to speak. Let me relaunch the poll. What was the result from your group? My picture is always in the way. All right. Almost there. 9 people answered. Let's see. There are only 3 choices. So if you're randomly guessing, you have 33% chance of guessing it right. 33.33 to be exact. All right, I will end the poll and share the results. When I typed yes, that's correct in the chat box, I was referring to the previous question. Okay, now this question. The correct answer is C. Okay, so 4 of you chose C. Does anyone from that group, from the 4 of you who chose C, want to explain why you chose C? So it's not me explaining all the time. Anyone have the courage to unmute and tell the class why they chose C? No? All right, I will explain it. What happens when you freeze water? Can someone tell me what happens when you freeze water? You fill up your water bottle to the brim, you close the lid, you put it in the freezer. The next day, what happens? It expands. That's right. Safi said it expands. Absolutely. Water is one of the unique property of water is it expands as it cools, right? Think about it. Generally, things expand when they're heated and contract when it cools, right? But for water, when you cool it between 4 degrees to 0 degrees, it actually expands. Okay, so that's what happens if you freeze a cell. If you freeze a cell, it will expand. The water in the cytoplasm will expand. The ice crystal will actually puncture the cell membrane. And then when you thaw it, you would end up having a cell that's like, you know, full of holes. So there is no more regulation. Things goes in, things goes out without any regulation. And as such, the cell will not change its size. Okay. You want to remember what I just told you because one of the lab questions involves understanding of this concept. Anybody have any question? You must have at least one question. Let's see. Would the cell just be doing nothing then? Yeah, pretty much because the cell is dead, right? It's just like a bag now, right? With a bunch of holes. So usually I get a common question following this, and the question is, so how come people can freeze their eggs, right? You have heard of that, right? Like celebrities, they want to freeze their eggs and then use it later. Right? You can freeze the sperm, you can freeze your blood cells. Anybody heard of that before? Probably you have, right? So how come that's okay? How come, you know, if you freeze the egg, you're not gonna kill the egg? Anybody know? Anybody thought of that before? Not sure, eh? All right, that's your food for thought this week. Okay, if you figure out the answer, you can send me an email. Right? If not, I will tell you next time. Okay, let's finish this. Choose the more concentrated solution in each of the following solution. So we have 80% water, 20% starch versus 90% water, 10% starch. Right? So the more starch you have, the more concentrated it's going to be, right? So that's the more concentrated one. 5 grams of sodium chloride in 50 grams of water versus 5 grams of sodium chloride in 25 grams of water. This is kind of like, you know, your regular grocery dilemma, right? Do you buy the 12 rolls of tissue of toilet paper for $7.99, or do you buy the 6 rolls for $3.99? Right? They are different amounts and different monetary value. How do you figure that out? Well, you have to figure out the unit price, right? Okay, so same thing here. One is dissolved in 50 grams, the other is dissolved in 25 grams. So maybe I can make this 50 as well. So how do I go from 25 to 50? You're going to have to multiply by 2. So we're going to have to multiply this by 2 as well, right? So now we can compare because they're both 50 grams, right? And obviously 10 grams of salt in 50 grams of water is more concentrated than this one. Last, you have 15% solute versus 25% solute. So the one that has more solute is going to be the more concentrated one. Okay, that's that. 3 types of bulk transport. So we have phagocytosis, and we have pinocytosis, we have exocytosis. We also have something called receptor mediated endocytosis. All right. And again, if you're not sure what these are, watch the recordings. Everything is explained there. Okay, long answer questions. Any specific one you want me to talk about? Please let me know and I'll do that. If not, I will just take up the application questions. Okay, so someone talked D and then the person requested 1. Okay, so let's do number 1. Sodium potassium pump. What is a sodium potassium pump? Okay, so this is your cell membrane. Cell membrane. This is the inside of the cell. This is the outside. Okay, so on some cells, you have this thing called the sodium potassium pump. The job of the sodium potassium pump is to pump 3 sodium out in exchange for 2 potassium. Okay, so that's one cycle. For each cycle of the pump, 3 sodium will be removed from the cytoplasm and 2 potassium will be brought into the cytoplasm. So over time, what's going to happen is because you keep on pumping sodium out, you will end up having high sodium on the outside and low sodium on the inside. Contrary, for potassium, because you keep on bringing them in, you would have low potassium on the outside and high potassium on the inside. Okay, so if you keep on doing this, you will notice sodium is being moved from an area of low concentration to an area of high concentration against its concentration gradient. And the same thing can be said for potassium, even though they're in opposite direction, but each of them is going against their own concentration gradient, which is why in order for this to keep on happening, you will need to have ATP. That's basically what the sodium potassium pump is. Now you might wonder, why are we doing this? Why are we moving sodium out and potassium in? It turns out you need to do this to keep your neurons functioning, right? And you will learn that in a lot more details next semester. All right. We have D and E for the other requests. So D is carrier protein and channel proteins. Okay, so both, how are they similar, right? The question asks you to describe similarities and differences, right? So let's do similarity first. Okay, so we're talking about carrier protein and channel protein. Channel proteins. So how are they similar? Similar. How are they similar? Well, they are both examples of passive transport. In other words, no energy is required and they are going down the concentration gradient. And they are considered as facilitated diffusion. And one more time, what that means is because the molecule is either too big, charged, or maybe it's hydrophilic, they are not able to cross the phospholipid bilayer on their own. And so they need a little bit of help from the membrane protein. Okay? So that's why it's called facilitated. The protein, the carrier protein or the channel protein is facilitating the diffusion that would have occurred if the molecule didn't encounter any barriers. Okay, so it's still going from high concentration to low concentration. So that's how they are similar. Okay, no ATP, no ATP. Okay, how are they different? Okay, difference. You can think of a channel protein as like a regular tunnel. Okay, so like this is your cell membrane. Okay, and then the channel protein would provide a passage, a tunnel, a channel for the molecule to go through. So the molecule, whatever it is, right? Maybe it's charged, maybe it's too big, maybe it's polar. It's just gonna go through the middle of the channel protein, and it will go inside. Doesn't have to go in; it could come out. Right? What determines which direction it goes is going to be the concentration gradient. If it's high on the outside and low on the inside, it will come in. If it's high on the inside and low on the outside, then it will go out. Okay, so the channel protein basically shields the molecule, whatever it is trying to cross, shields it from the hydrophobic environment created by the phospholipid bilayer. Okay, so it's like a tunnel. Okay, you go through a tunnel. On the other hand, the other one, what is it called? The carrier protein. Carrier protein is it's like Pac-Man. Okay, so the molecule will bind to the carrier protein, and then what's going to happen is it will grab onto it. The protein will grab onto the molecule, and then it will change the shape so that it's now opening on the inside, right? I don't think I can find the same color, but whatever. And then it will split it out like this. Okay, so it binds, it causes a conformation change in the channel protein, in the carrier protein, and then it's gonna spit it out. So if an analogy for a channel protein is a tunnel, an analogy for the carrier protein would be like a revolving door. Okay, so like you know those ones that you go into, like a building. I don't know if you know what I'm drawing. So you get in one side, you push the door, and then you end up on the other side and you exit, right? So this is kind of like that. So the difference is how the molecule gets through. Okay, this one, the protein does not do anything. The membrane protein, it just provides a corridor for it to go through. This one, the protein actually grabs onto the to the thing trying to cross, changes the shape, and then spit it out on the inside. I hope that answers your question. Phagocytosis and pinocytosis, I'll just verbally discuss this one. They are both examples of endocytosis. Okay, so endocytosis is a type of bulk transport. Bulk transport is B-U-L-K, bulk transport. It means you're moving large quantity of substance into or out of the cell. Okay, so when it's phagocytosis, you are engulfing something. A perfect example of this would be your white cells eating a bacteria or something, and that would be, bringing the bacteria in, fusing it with the lysosome. That's an example of phagocytosis. All right. Pinocytosis, it's essentially the same thing, but you are not bringing in like a physical object. Okay, you are bringing in mostly fluids. Okay, so some of the cells in the kidney, they do that. Okay, they do pinocytosis. They bring in large quantity of fluid into the cell, and that's why it is called cell drinking as opposed to cell eating. Okay, so that's how they are different. They are similar in the sense that they are both bulk transport and requires ATP. Okay, let's go through the application question here. In 2007, a woman who competed in a radio station contest to see how much water she can drink without going to the bathroom died of water intoxication. Explain why drinking a large amount of water in a short period of time could result in water intoxication. It's quite straightforward. You should understand this concept quite well by now. If you drink a large quantity of water, it's going to dilute your blood. Okay, it's going to dilute your blood. So let me write down for you. Dilute the blood. So now you create a hypotonic environment for your cells, for your blood cells. Right? So that could lead to lysis, right? So your blood cells is gonna burst, and if your red cell starts to burst, then you won't be able to distribute nutrients and oxygen anymore. So Makia asks, how much water would be unsafe? I mean, it has to be quite a bit. Like you would feel sick to the point that you don't want to drink anymore, and then you force yourself to drink more. It has to be that point. Like, you know how some people say, my mom does that actually. Like she would drink 8 cups of water, right? That's fine. That's not gonna create hypotonic. Right? You will have to keep on drinking, and you're like so sick of it, and you force yourself to drink more. Then that's when it's like, dangerous. Another thing is when you dilute the blood, then you are also diluting the electrolytes, right? So like calcium, magnesium, all those things are important. Okay? If you dilute the calcium concentration, then calcium might not be able to do its normal function. You need calcium for many things. For like clotting your blood, you need calcium. I'm talking about like calcium 2+, right? The ion. And you need calcium for muscle contraction, right? So your muscle might become weak, and if your heart muscle don't have enough calcium, they might not be beating either, right? And that's the sort of stuff that would kill you. Now, if a person is suffering from water intoxication, but like not dead yet, right? One of the ways to bring them back is to give them like a hypertonic IV solution, right? Okay, so you try to rebalance. You want to rebalance their blood concentration. Next, single cellular organisms that lives in fresh water are consistently exposed to hypotonic solution. Speculate what kind of transport mechanism would be used to prevent lysis. So last class we talked about this thing, right? I don't know if you remember it. There's a mouth here, and then it has a lot of cilia around it, right? I think some of you mentioned the name of this. This is the paramecium. So paramecium lives in pond water, which means it is always going to have water coming in. So if paramecium don't do something about it, they will burst and die. But they don't die because they have a special vacuole, okay? And there are some like contractile fibers around it, kind of like mini muscle, if you would. So that would suck up all the water from the cytoplasm and then pushes it out. It's like it's having its own internal plumbing system. Right? So by actively pumping all the excess water out, it makes sure it's not gonna burst. Okay. Marine animals have another problem, the opposite problem. They are always in a hypertonic environment, right? You think about like a dolphin or like a fish, they live in these salty environment. They are always at risk of losing water, right? And so they need to have other mechanisms to cope with that. And we'll talk more about it again next semester in the next course. if you are in my class again. Next, salting has been used as a method for preserving food. Coating meat with layers of salt will dehydrate the meat as well as any microorganism. Explain how this process works. It's quite obvious. By having a lot of salt, you are creating a hypertonic environment, and it will suck out all the moisture from the microorganism, drying them out, killing them. Without microorganism, they will not spoil the food, and the food will last longer. But of course, like salted food is not. It's not the most healthy food because of the obviously it's very, very salty, and having high sodium intake can causes its own set of problems as well. Okay. Any follow-up questions? Good. Okay, let's fill in the blanks. The plasma membrane consists of 2 layers of phospholipid bilayer, cholesterol are important for maintaining proper fluidity. Okay, too much, it's gonna be too leaky. Too little, it's gonna be hard like a wall. Nothing goes through. We need to have the right amount. Break down molecules into smaller units. That is catabolism. Building molecules, on the other hand, it would be anabolism. And ATP molecules contains adenine, as in the nitrogen base from DNA, a sugar, and 3 phosphate groups. Diffusion of water is called osmosis. And we just talked about this. Carrier protein, channel proteins are facilitated diffusion. Again, no ATP required. An animal cell will shrink when you place it in a hypertonic solution and has lower solute concentration than the inside of the cell. That would be hypotonic. Cell eating is phagocytosis. Cell drinking is pinocytosis. And if you put a cell in a hypertonic solution, it will undergo plasmolysis. Okay. That is that. This one is just, you can fill it in yourself. I think I already filled this in the lecture, probably. All right, so next week you will have a quiz on last week's material on the cell organelles and stuff like that, and then you will also have the quiz will also include what we've learned today. Okay, we have about 10 minutes. Let's do this Kahoot thing, and we will. It's like a review. I don't know if you played this with your other teachers yet. Have you played this with your other teacher, Kahoot? No, not sure what it is. In high school, yeah, that's great. Okay, so let's see. What you do is you will open a browser, or like you can do it on your phone, and oh my goodness. There we go. Okay, I'm gonna turn off my camera. So you type this in, okay, www.kahoot.it. I will paste it in the chat room. And then when you're there, you can key in the pin. Just right here, I'll copy the pin as well in the chat room. And then you can make up a name for yourself, as long as it's an appropriate name. How do you even do that? How do you even like put a icon? That's so cool. And now you're just showing off, right? with all these icons. Oh, okay, you're doing it on the phone. I see, I see. Wait a little longer in case people want to join. For those of you who haven't played this before, it's like, you know, those pre-game show in the cinema, right? Remember when we used to go watch movies? Seems like so long ago. Okay. Is anyone else trying to join? Should I wait? Or like, I mean, we have like what, 8 more people? Are you trying to join, or like, you don't have to install the app. If it tells you to install the app, you just like use the web version. It's fine. Okay, I'm gonna get started. Otherwise, we'll never finish. If you want to join, you can still do that, I believe. So here we go. You can also just do it in your head. It's fine. The rough ER is the correct answer, right? Not the lysosome. Lysosome are the little vesicle that, you know, bubbles off from the Golgi bodies, right? Okay, the one that is directly connected to the nuclear envelope is the rough ER. Next one. The smooth ER is where you make the lipids, right? Ribosome, they make proteins. Ribosomes do not make lipids or cholesterol. Okay, so that's why it's not the first one. You cannot see the question. Some of you say you cannot see the question. You cannot see the question on your device. You have to see the question from my screen. I believe that's how it works. So, you know, yep. So we specifically talk about this in last week's tutorial, right? Detoxification involved the smooth ER. The lysosome, they break down lipids, they break down carbohydrates, and then they break down all organelles as well as you know, like things that the cell engulfed, right? But the detoxification happens in the smooth ER. Cell recognition, this is what we learned today, right? Phagocytosis is if you actually eat them, okay? This is talking about recognizing it, right? It's this one, right here, right? This one, right? Cell recognition. Next. Hypotonic solution, right? That's the one where the water comes in and then will cause it to burst. Hyper is shrinking. Seawater is basically a hypertonic solution, right? Which of the following requires the use of ATP? Yeah, phagocytosis. Okay, diffusion is just movement of water. There is no energy required. Diffusion from high concentration to low concentration, no ATP required. Facilitated diffusion, we just talked about it, right? Remember, like in the fill in the blanks, right here. Look. Facilitated diffusion, no ATP required, right? So it's just using the channel protein to help, right? So phagocytosis is the only one in this list that requires the use of ATP. So which of the following does not affect the membrane permeability? Yep, the size of the cell. So let's talk about the other one. pH will definitely affect it because there are many proteins in the cell membrane. And we learned in lecture 2, if you change the pH, you will change the way the proteins are folding. So it might misfold the cell and cause the membrane to be unraveling. Temperature, if you increase the temperature, then the fluidity is going to increase and the cell membrane will become more leaky. A membrane composition, well, it depends what the membrane is made up of, right? If the membrane has more cholesterol, for example, then it's going to be more permeable. If it has more proteins, then it might also be more permeable compared to less proteins. So the size doesn't matter. You can have a really big cell and a really small cell. If the membranes are made up of the same stuff, then it's going to have the same permeability. Just 3 more questions. Yeah, that's good. Outer membrane and the inner membrane, right? The inner one folds into the cristae. We labeled the mitochondria before. Lysosome only has one membrane, okay? Yeah, false is the correct answer. Chloroplast is not unique to plant cells. They are not in animal cells for sure. But last week I told you, you can find chloroplast in bacteria or you can find chloroplast in some of the protists, right? So definitely not unique to plant cells. But it is found in plant cells, but not animal cells. And the last one. Centrioles are found only in animal cells and not plant cells. Okay, centrioles are the ones that are arranged themselves perpendicularly and create the spindle fibers to move the chromosomes during cell division. And that is it. Hopefully that gives you an idea of where you're at and how much studying you will need for the next week. So just to reiterate, you will not have class next week. Next week is just for you to do the lab as well as prepare for the quiz. We will not be meeting in a synchronous session next week. If you have time, then you can start watching the lecture 5 recordings. Otherwise, the week after is engagement week, also no class. So our next synchronous session will be on the 22nd. Okay, of course, for your class, it's going to be like 12:30 to 2:30, right? If you need help with the lab, feel free to contact me. Otherwise, good luck with the quiz and have a good weekend, everyone. See you all next time.